Welcome to this Morphit demonstration. My name is Ted Hawkins and I'm from the Edge Software Consultancy, and today I want to show you how we can use Morphit to run an in-vivo behavioural assay. In this case, it's the assessment of a compound's ability to reverse stimulated behaviour. The procedure is fairly straightforward. The first steps are to define the experiment protocol and the treatment groups, and the next stage then is to bring the animals, the pool of animals that's available, and assign them to the treatment groups. We can then randomise the assignment and the treatment groups, and then make the scoring sheet available for the technician or the scientist who will actually capture the data. As the data is captured, then it is transformed, analysed and presented graphically and in a results table. We'll start the demonstration by creating a new binder from a template. File, New from Template, and select the template that we're interested in. This will open a brand new workbook. In here we can look at the sheets in the workbook. And we also see an instructions panel describing how we should run the assay. So let's go straight to it and define the experiment protocol. Okay, so we can select the species from an internal dictionary, and the strain is linked to the internal dictionary as well, so we'll only see the strains of that rat. So once that's completed, we can mark it and move on to the next section, which is to define the treatment groups. So on this page we can see that we have 3 treatment groups already in place. Each treatment group has a role: negative, positive test, and each of these roles has some type of substance or compound associated with it. Each will have its own dose, route of administration, cofactor dose, cofactor and route, and also has a collection of animals. The first thing we need to do is add enough treatment groups. So I want to have 3 test groups at different doses. So we can either go through this right key menu here or press Ctrl-I. By selecting the role, we'll bring in the compound automatically. We'll enter our doses and routes of administration, cofactor, and its route of administration. I also notice that the first test group has got more animals than I need, so I'll remove that animal. And we'll add some more animals to the control groups. Okay, so now we have 2 control groups and 3 test groups. With the test groups defined, the next stage will be to bring in the animals. Now, in the usual process, we'd order the animals, we'll get a batch of animals back, usually more than we need. There'll be a pool of animals that we'll assign, usually randomly to the treatment groups. So assuming that there's a data file containing the details of the animals that have been dispatched, we'll import that. And there already is a file associated with this, but we'll just overwrite it. Okay, so this is, in this case, a very simple text file. It's comma separated. And we're going to select the columns that match the data. Now, as we said, there are more animals dispatched to us than we actually need. The animals are automatically allocated to the different treatment groups, and those that aren't allocated, you'll see, have no treatment group assigned in the use column. We've created a spare animals table so we can look at create a list of animals that have not been used. Okay, there is one final randomization that we do, which is to randomize the order of the treatment groups and re-randomize the allocation of the animals to the treatment groups. And that's done with a single button, although we could split these 2 steps. We can see by pressing it that the animals have been reordered. So the treatment groups have now been set up, the animals have been assigned and treated, and at some point the scientist or technician will need to gather scores. So we can see here the treatment groups have been organised randomly. There's a full treatment description on here. Now, of course, we could just have put through a random number so people couldn't see the actual treatment. The animal code is in here, and I've already entered the weights. The usual the usual next step would actually be to print off this file onto paper and take this paper into the lab and write the scores down. The scientist would then come in and transcribe the data into the system. Okay, so we've moved the data in. Now it may be that we see an outlier in this data, and one thing we can do is simply knock that data point out. So it doesn't delete the data, it just marks it out from the analysis. So with the data entered, the data is processed as it's entered, and we can see the results in the reports table. So here we can see a very strong response that varies with dose for the test compound. However, the weight of the animal doesn't. And below we see the initial body weight, so we can see that there was no bad distribution of animal weight into each groups. We can also see the results in a table. In this case, we've repivoted the table again, so it's easy to digest. And this may be printed off, but we can also save this as a CSV file. We can open that file. And we see a nice flat structure that's very suitable for loading into other applications or databases. So that's the assay run. This document will be saved on the file server or in your ELN, and the experiment's now complete. If you'd like to download this example, it's available as a member download from our website, as is the full documentation on how to build it. Thank you very much for your time.